I once heard a story about a graduate student who predicted that the breeding protocols of laboratory rodents would compromise the laboratory system in terms of its relationship to so-called wild type versions of the same species. So you have the bred rodents and you have the wild rodents, and that they would be distinguished by virtue of the fact that the non-coding nucleotide sequence at the end of the chromosome, known as telomeres, would be wildly different in length if the prediction were true from pure evolutionary theory.
Wow.
Yeah.
Yeah. That story, it didn't happen exactly the way you said it, but, you know, it's been a lot of years and takes a second to get back there.
Yeah. I mean, it's you did that.
Yeah, I did that.
And that story, unfortunately, has not really been told. And it is, in some sense, your central origin story as a biologist.
It's a pretty good one. And it definitely changed the way I saw myself in a way that has been very productive.
Okay. I want you to talk to me about that story. And I will, because I lived it with you, I know that it happened, and I know that it got buried.
Yep.
And I know that it's part of what I'm calling the distributed idea suppression complex, because quite frankly, you were not the only person who was a part of this story, and the story had to die because it said something.
So when I went into the literature, what I found was that people had played around in the neighborhood, but that there was a particular fact which blocked every attempt to make sense of what was going on. And the fact was that rodents were understood to have ultra-long hypervariable telomeres. And I didn't know what that meant at first, but the more I looked into this possibility, the more I realized that dozens of long-standing problems would be solved if my hypothesis was true, but that my hypothesis couldn't be true because basically mice have long telomeres and short lives. Why is that? And I banged my head on the table for a couple weeks trying to figure out what was going on. Yes, maybe even literally on occasion. But the question was, I began to wonder if there was something wrong with the idea that mice had long telomeres. Sometimes, like in Hayflick's case, it turned out that a bunch of people were copying some wrong result in it, so it seemed like a lot of people had seen it, but only one had. And I checked, was it true that there was that everybody was parroting one study that said mice had long telomeres? And it turns out lots of people had tested it. Mice have long telomeres, like 10 times the length of human telomeres. It just didn't fit. So finally, it occurred to me that it was possible that what was going on, I discovered something in trying to figure out what they meant by mice, right? There's a lot of species of mice, but all the mice that we use in the lab, with rare exception, are from one genus and often from a particular target species.
You were focused, if I recall correctly, on Muspretus?
No,
Mus musculus?
Mus musculus, which is the common one.
Yeah.
What shocked me was that it turned out all the mus musculus that were being used in labs across the country, and in many cases farther afield than that, were coming from one place, which I had no idea. There was one.
I remember getting a phone call when you said, "What do you know about the Jack's lab?"
The Jack's lab in Bar Harbor, Maine, right? They seem to be the source of everybody's mice. And so it began to be, it was a possibility I could not shut down in my mind, that there was something about what was going on at the Jack's lab that had resulted in the mice that were being sent out to all these other laboratories.
As if they were representative animals.
Right. These are model organisms. People were just using mice because mice were a convenient mammal. But they're all coming from one place. And it began to occur to me that that one place was not just a source of mice in the sense that we might think it. It was actually a selective environment that was impacting those mice. And when I dug deeper, it turned out that the mice they were descendants of a long lineage that had lived in captivity under conditions at the Jack's lab. And at some point, I realized that the most likely thing going on was that there was something about this environment that had wildly elongated the telomeres of these mice. And that was simultaneously an unbelievable idea, but the only one I could think of that made sense of everything I had seen. And so,
well, it's unbelievable because the consequence, I mean, look, I have not even heard whether anyone has said, yeah, we did that, we screwed that up. But it is like your favorite model organism from mammalian trials being screwed up by a central facility, because also there's this weird thing where medical people very often stop taking into account evolutionary theory because they treat that as, well, that's that class I took in college or the beginning of graduate school.
Right. So I began to focus on this question, and I did something that was the right thing to do, but I did it in a way I will forever regret. I found somebody who was represented in the literature who I regarded as very well-versed. They made sense to me, their papers. Her name was Carol Greider. Carol Greider is now a Nobel laureate. She was not at the time. She was the co-discoverer of the enzyme telomerase, which is the enzyme that elongates telomeres when that occurs.
With the famous and co-Nobel recipient, she was the student of Elizabeth Blackburn.
Elizabeth Blackburn, exactly. She was her student, and they shared the Nobel Prize with Shostak. In any case, her work seemed good to me. I called her up cold. I went into the insect division office, and I sat down at the phone. I called her. I said, Carol, you don't know me. I'm a graduate student at Michigan. I'm an evolutionary biologist. I'm racking my brains trying to understand something. Can you tell me, is it possible that mice don't have ultra-long telomeres, that it's only laboratory mice that do? And she said, that's really interesting. I'm pretty sure that mice have long telomeres universally, but it is odd that if you order mus spretus instead of mus musculus and you order from European suppliers, the lengths are very different than what you get if you order mus musculus from the Jack's lab. And I said, whoa. And she said, yeah, that's really interesting. And then she said, I can't remember if it was the same phone call or if we had a second phone call, but she said she was going to put her student, her graduate student, Mike Heeman, who I think is now at MIT, on the project, and he was going to do a little work to figure out whether there was anything to this. And Mike did some work. They sourced some different strains of mice that were actually not wild mice. Wild mice would have been the right test, but she couldn't get wild mice for obvious reasons.
You have to go out into the woods.
Right, exactly. And so she got several different strains of mice that had just been in captivity much less time. She actually got one strain of mice that was treated very differently in captivity, but never mind. She put her graduate student on it. He measured their telomere lengths. And I get this excited email. Mike Heeman sends me an email that says, effectively, whoa, the hypothesis is true. Mice have short telomeres.
Because it's a breeding protocol that is the alteration in the evolutionary landscape for these laboratory mice, and because it's acting on a non-protein coding region, the adaptation to a change in the breeding protocol can be extremely rapid. It doesn't have to undergo some sort of completely crazy, typical Darwinian story about random mutation and some of them being retained and others being rejected.
It's even better than that. The creatures are presumably, so we haven't gotten to what the breeding protocol has to do with this, but the creatures are built in some sense to detect how dangerous their environment is. And to the extent that the level of extrinsic danger changes, their telomeres respond quickly so that they are better adapted to the environment. So they're built to detect the environment. And then what is actually a strict matter of market forces.
Okay, so there are no predators in this environment.
There are no predators in this environment.
And we're not killing them particularly early based on their skill. So environmental insult is sort of absent.
Environmental insult is more or less absent. What we are doing is imposing an economic rule on breeding so that we can maximize the rate at which we turn mouse chow into mice, which is obviously economically the right thing to do if you're selling mice to all these labs. You want to produce as many mice as cheaply as possible. So, okay, so let me clear up why the breeding protocol. And I should say, That it is the breeding protocol that is causing this, that part, I would say, is still a hypothesis. It has not been directly tested by anybody. But what I would say is many hypotheses were tested in the aftermath of the discovery that lab mice have bizarrely long telomeres and wild mice don't. And no other hypothesis has stood up to scrutiny. So it is the last hypothesis standing, and I'm all but certain that it will turn out to be true.
Yeah.
The reason that the breeding protocol has this weird effect is that when you throw out the mice at 8 months of age, you eliminate selection against cancer, you turbocharge selection in favor of...
So when you throw out the mice for breeding purposes at 8 months of age. Okay.
You throw them out for breeding purposes at 8 months of age, you are increasing the importance of their early life breeding, and you are discounting anything related to their ability to fend off cancer because they don't live long enough in that period of time to get cancers that kill them. And so what has happened, according to this hypothesis, is that the mice that have longer telomeres have driven out the other animals from the colony. The trait of having long telomeres has swept through the colony, and the telomeres have been elongated to an absurd degree, creating animals that do all die of cancer. And interestingly enough, another thing that's evident from the literature is that if you look at their tissues do not age in the way that a normal mammal's tissues age. They remain young.
Well, so there's one aspect of aging, but that there's a far darker interpretation of what you've just said. If I'm understanding it, correct me. I've never taken a class in biology, but I've lived this adventure with you. Those tissue have, at a histological level, at the level of how cells are organized, the possibility of radical histological repair.
Yes, radical, effectively indefinite capacity to repair, which is going to come back in this story in the worst possible way. And I have purposefully not shown Carol Greider the paper in preparation, which I am afraid she might have read some way. The reason I didn't show it to her was because I wanted to preserve her independence as a reviewer for the paper. I was hoping, because I still thought she was an ally of mine, I was hoping that Nature would send it to her to review and that she would look favorably on it, especially since it was, very clear that she had done...
It was her lab that made the confirmation.
Yeah. And another thing I forgot. I asked her at some point, something that now rings in my ears. I asked her, Carol, you've now got this result about, no, actually lab mice have long telomeres, but wild mice have short telomeres. That's a big result.
It's a hell of a delta.
Where are you going to publish it so that I can cite it in my paper, which is the natural thing to do? And she says, we're not going to publish it. We're going to keep the information in-house. That was her phrase. I was too young to understand what the hell she was talking about.
I'll be honest, I'm 54 and I don't quite understand it myself.
Well, it's so heartbreaking. What she has effectively done is decided I could publish this result.
And then everyone would have it.
Huge. But then I'm on a level playing field with everybody else. If I don't publish this result,
I have a stream of papers I can get at.
Then I can start predicting other results. Nobody will know how I am doing that thing. I will look like a super genius. And so holding it in-house is a mechanism for a whole slew of papers.
To be 100, you can afford to bend over backwards and not make inferences. Let's say the following. Holding it in-house is a seemingly inexplicable decision in science, but for the fact that it fits at least one story of this kind, which is that it is consistent with wishing to publish a stream rather than the source of the information that would allow you. So you can either do one discovery or you can do a stream of predictions. And that makes a certain amount of sense given the ruthlessly competitive grant winning environment. And we don't know exactly what happened, but there is no world that I know of in which you're allowed to hold back that kind of information because in part of what's on the line.
Right. So,
I mean, this is not just a question of academic interest.
No,
because these mice are used for medical testing purpose.
Not even that. It's medical testing, but it's also all of the science relative at least to cancer, senescence, wound healing, all of the science that is stacked on these mice that is contingent on their function relative to their telomeres is all compromised.
Right.
You're letting year after year of this stuff accumulate. It's malpractice at an incredible level. So I don't know that she has turned on me, but I call her up and I say, Carol, we are stunned to find that our paper was turned away without review from Nature.
Without review.
Without review. We need your help. Can I send you the paper and have you look at it? And she says yes. And I send her the paper. And she sends back the paper with an unbelievable number of intense criticisms that are not sensible. She pans the paper, does not believe a word.
Do we still have the copy?
I have that paper. I have that paper with her handwriting. I believe I also have the FedEx envelope in which she sent it to me. But so the paper was accepted by Experimental Gerontology. They went on to publish it.
This is called Life Slow Fuse?
No, Life Slow Fuse was the title as sent to Nature, and I changed the title because I did not want to compromise the story. I didn't want to confuse the story.
The original submission was called Life Slow Fuse.
Right.
We probably have a copy of that somewhere.
Of course.
All right. Then the experimental gerontology paper, what is it called?
The Reserve Capacity Hypothesis.
That's
okay. Which is a much less catchy title, but nonetheless, the paper, I'm very proud of how it's written. People read it who were not expert, could understand it. The abstract is extremely clear, and it ends with the clear point that because we have unearthed, we have predicted, and Carol Greider has shown that wild mice telomeres are short and that telomeres have been elongated by captivity, that there is a clear danger that the mice we are using for drug safety testing are biased in an egregious way. And the bias would look like this. A mouse that has very long telomeres has an indefinitely large capacity to replace damaged tissue. And it has a vulnerability to cancer that is preternaturally high. So we may be overrating, if we use these mice, we may be overrating the danger of causing cancer and vastly underrating the danger of toxicity. And in fact, one of the things, so the point was, you give a mouse who's got an effectively infinite capacity to replace its tissues a toxin, and either the toxin is so deadly that it dies right away, but if it doesn't die right away, it just eats up the insult. So those animals would lead us to release drugs.
By insult, what you mean is cellular necrosis?
Damage. Yeah. What this would cause us to do is release drugs onto the market for human use that are highly toxic across the body. And
wait a second. If the mouse standard was the last standard,
well, no, even if it's not the last standard,
well,
because
it's important to say this.
The problem is, I mean, you can imagine how hard it is to test on large, slowly reproducing animals.
Well, and the ethics of testing on humans is very
absolutely
restricted. So mice is the last cheap place.
It's the last cheap place.
Large N data.
Not only large N, but it's the one place that you can make the following move. You can imagine that in many circumstances, the accelerated lifespan, the accelerated life cycle of mice allows you to see long-term damage as it would accrue in humans on a very short time scale. That doesn't work with monkeys. It doesn't work with human patients. It works with mice, maybe. But in the case of mice with ultra-long telomeres, those insults will be invisible.
Well, I want to back up because I think this is a really important part of the story. What you're saying is if you take an organism that has an expected, let's say, 40-year lifetime, it's very expensive time-wise to say we ran this experiment and found that there was no immediate damage that was visible, but towards the very end of their lives, we saw a marked increase in morbidity. Yeah,
I mean, if you took a drug and it knocked 15 years off your life on average, that might not show up in any notable way in a short-term study.
There was pressure to...
Right. And nobody is going to want to let drugs, you don't want to wait 40, 50 years to find out what happens to these patients. So what we do is we make the assumption that if we give large amounts of a drug to an animal that lives a very short life, we will see those effects early. And if the animal happens to have ultra-long telomeres, you won't see those effects early. So it's a perfect storm for causing us to release drugs that should never have been released into public.
Can you think of one?
Oh, I sure can. Vioxx, for example. So Vioxx was discovered to do heart damage, right? Heart damage. Why do we know that it's heart damage? Well, the thing about hearts, hearts, for reasons we can get into maybe another time, hearts have a very low capacity for self-repair, right? That's why they're vulnerable to heart attack.
Turnover.
Not much capacity for repair, not much turnover. Now, there's an adaptive reason for that. But hearts don't repair themselves very well in a healthy person. And when they fail, it's hard to ignore, right? If somebody who's 30 has their heart fail, there's questions asked, right? So anyway, Vioxx was released into the public, having passed drug safety testing.
This isn't the only system that doesn't have a lot of mitosis. Like, for example, neurons.
Neurons don't have a lot. Cartilage doesn't have a lot.
Got it.
Your eye cells don't. Now, note, all of the tissues I've just mentioned, when was the last time you heard about anybody having, cancer of the cartilage of their knee, cancer of the heart?
No, if they get brain cancer, it tends to be glial.
It's glial cells, exactly. Right. So the tissues that have very low capacity for self-repair do tend to wear out, and they don't tend to get cancer, which is exactly one of the predictions of my paper.
Right.
Okay. So Vioxx is known to do heart damage. That created a big scandal because how the hell did it get through drug safety testing? Turns out a lot of drugs have done this. We've seen it in Gleevec, fen, erythromycin. Your doctor probably still doesn't know that erythromycin does heart damage.
Yikes.
Right. There's all of these cases of drugs that were released and then later understood to do heart damage. Now, my claim is they don't actually do heart damage. They do cellular damage, and the heart damage is the only conspicuous.
Thing. Yeah. This is
at the point that my paper is out.
Right.
And it very directly alleges the danger with these drugs being released when they're not safe. And the drugs have started emerging and turning out not to be safe. Right. And the government is now really interested in what's going on. The government puts together a FDA commission to study the question of the book that they put out. Literally a book that they put out at the end of their study is called The Future of Drug Safety.
It's a blue ribbon panel.
It's not exactly clear what it was. What is clear is that you can search the manuscript of this book. Nowhere does it mention mouse.
Antagonistic
pleiotropy. It doesn't mention antagonistic pleiotropy. It doesn't mention the genus mus. It doesn't mention telomeres. It's not in there. It's alleged in the literature in broad daylight that this is what is causing the problem. And
well, now this is the vampire effect where you don't exist if nobody reacts.
Right. And so I start going to members of the press. I think this is a huge goddamn story. Somebody is going to make a career on it. And I call up members of the press, and it's always the same, right?
Always the same.
It's always the same. They're very excited about this story.
They're initially, the reporter.
The reporter is excited.
Yeah.
And then the reporter
talks to someone.
To someone, and then either they stop returning your calls or they say, I'm sorry, the story doesn't hang together. It's again and again and again. So I have to go through the final Carol Greider chapter in order for this story to fully make sense.
Where the Nobel Prize is given?
That's the very tail end.
Make sure you include that.
Okay. So at the point that my relationship with Carol is changing its tenor and she is becoming hostile and I'm not clear on what's going on, I contact her and I discover through talking to her that she and Mike are about to publish their paper on the long telomeres of laboratory mice.
So this is the delta between wild type and laboratory mice.
Yeah. And I'm shocked because she's told me they're keeping it in-house. And Instead, they've got a paper that she says, in final revisions. They are that day submitting their final revisions to nucleic acid research with their paper. And I say, Carol, can I see the paper? And she says, yes. And she sends me a manuscript, not the preprint of the paper. She sends me a manuscript of the paper, no acknowledgments, no figures. And I contact her and I say, can I see the acknowledgments and the figures? She sends them to me. And I contact her and I say, Carol, I'm disturbed. This was my hypothesis that you were testing. I should probably be an author on this paper, but at the very least, I need to be an acknowledgement in this paper so that I can go back and point to it and say
it changes everything, that it was a prediction. It wasn't just something that was stumbled upon.
Absolutely.
Yeah.
And her response is, I have been through my email and I see no evidence of the communications you are talking about. Now, when I said at the beginning that
called her,
I had called her. Carol does get awarded the Nobel Prize. Carol Greider, Elizabeth Blackburn, and Shostak. Shostak, who mentions at the point that the Nobel Prize is awarded that he was shocked as all hell to get a Nobel Prize because his work was so deep in the history of telomeres that we just didn't expect it. And suddenly...
No, I should say, I want to be very clear, right? Yeah. All of these people have made fantastic Nobel-worthy discoveries. There's zero allegation that these people
weren't deserving. No,
absolutely.
No, and they, Carol and Elizabeth got their Nobel Prize for the discovery of telomerase, which is a huge progress. So anyway, I don't deny that they were worthy of this prize, but what Carol Greider does with her Nobel lecture, right? Nobel lecture being the biggest lecture a scientist will ever give, the lecture that...
And filmed,
is she delivers a paper in which she very oddly has now embraced my entire set of hypotheses about the effect. She has come over from the comparison between the paper of mine that she panned and said didn't make any sense. She is now a total convert to the idea that senescence across the body is being caused by Hayflick limits that are telomere-based.
Okay, and this is the first public incident that we know of in which the delta between the negative comments on your paper, which is not an anonymous peer review. We have that.
We have it in an envelope from her.
Got it. And it's immediately after the Nobel Prize that the wisdom of that line of thinking is embraced.
Right. But there's more to the Nobel lecture. So she spends her Nobel lecture on what is admittedly a very beautiful presentation of the connection between telomeres and senescence. She goes through tissue after tissue, says cirrhosis of the liver is what happens when you have short telomeres in your liver, etc. She goes through tissue after tissue. She projects the data, the blot actually, from the paper with Mike Heeman, the paper that I should have been a co-author on.
Yeah.
She projects it on the screen. But she does some weird freaking dance where she, instead of describing the long telomeres of laboratory mice as a major bug in the system, she describes it as a happy accident, effectively, because it allows us to test certain things. Like, oh, isn't it delightful that they have long telomeres? And it's like, what the hell are you doing? There is so much writing on correcting this, and you're presenting it like it's just a bonus. So she goes through this. There's no mention of me. There's no mention of the actual implications of the long telomeres for things like science and safety testing and all of that. And I can't seem to raise the issue of the safety question with anybody, right? At best, I get journalists who are interested until they call somebody, and the somebody's on the other end, I know what they say. They say, everybody knows that mice aren't great models. In fact, there's a paper out there that says something like, the mice lie. It's not about this issue. It's just about the fact that mice aren't a perfect match. The issue in question could be solved. It could be addressed thoroughly. And for all I know, Once the Jack's lab figured out what they were doing, for
could change
all I know, they
protocols.
quietly have fixed this. And there was I've heard that there was a private meeting in which they decided.
Well, look, this is the thing.
Yep.
You see something like this in statistics. Everybody knows that most distributions that are bell-shaped are not normal, right?
Yep.
And on the other hand, we all use normal distributions. Yep. And as a result, there are lots of things that at one level everybody knows,
Yep.
but don't percolate down to the important layers in which we test things. And I don't know where, like you and I have never been able to fully put together, because we're not molecular researchers and I'm not even a biologist. How important are these results? How robust are they? Has there been a change? This is a quiet world at some level.
It's a quiet world, but I think what I have concluded, working backwards from the phenomenology of the field and how it reacts to this problem, is that there's a tremendous amount resting on failing to acknowledge the error, even though the error was obviously an honest error to begin with. They would rather sweep it under the rug. I mean, imagine you've got all these knockout mice, right? These knockout mice, there's a major investment in them. It takes a lot of work to knock out a particular gene.
You've got a single point of failure,
right?
Whose projections are tendrils into everything,
right? And you've got how many careers built on papers that are now suspect.
This is like an